Hi everyone. So we're going to continue what we started with the meiosis and genetics connection. And again, please add this as your final component to your Cornell notes in your notebooks. So what we talked about last time is a form of cell division called meiosis. And the whole point of meiosis is to take us from a full set of chromosomes, in our species 46, to a half set of chromosomes, which in our species is 23. And then sexual reproduction, each biological parent, plant, animal, doesn't matter. One biological parent contributes a half set, the other biological parent contributes a half set, and then the offspring receives a full set. In our species, it's 23 from the biological father, 23 chromosomes from the biological mother, for a total of 46. So that's the cell division part of it. So now what we're going to do is link that to the process of cell division. So when we've been doing genetics and we've been writing genetics problems, we would write our reproductive couple like such, right? And then let's just say that they are both heterozygotes, okay? So let's use big R, little r. If you like R's, use it. If you don't like R's, use a different letter. Totally up to you, right? And then we then said, all right, when these two reproduce, the biological mother contributes either a big R or a little r. So she separates her alleles. Hey, guess which process separates those alleles? Right here, that's meiosis, right in there. And then here is the same thing, the big R here, the little r here. And so what process separates these alleles? You guessed it, meiosis, okay? So when you look at the biological mother here, let's zoom in on her. And we said that she is big R, little r, she is a heterozygote. What does that literally mean? Well, she carries a pair of chromosomes in what we call a homologous pair. This is a homologous pair of chromosomes. A homologous pair. This is actually redundant. Logos means pair, homo means same, so what this literally means in Latin is same pair. And so when we say a homologous pair, what we're really saying is it's a same pair, all right? But no one says just homologous, it's a homologous pair. Fine. So if she's big R, little r, what that literally means is there's a gene that sits here, and there's a gene that sits here, and this gene codes for the dominant R allele, and this gene codes for the recessive R allele, right? And then when these genes are expressed, that's transcription and translation. So what happens then is this mother carries both chromosomes in all of her cells. And then when her ovaries go through meiosis, she then produces ova that either offers her offspring the dominant R or the recessive R. And this step right here is meiosis. It's the separation or segregation of chromosomes. Now, usually segregation has a very negative connotation because people talk about racial segregation, and that is ugly. That is separating people based on their race. It's horrible. Yet in biology, the segregation of chromosomes is a fundamental concept. We have to segregate our chromosomes. We have to separate so that we only offer our offspring a half set. So this is a full set here. This would be a diploid cell. These are only half sets, therefore this is a haploid cell. And this is a haploid cell. Okay? So what we need to do is just like when we drew mitosis, there was prophase, metaphase, anaphase, telophase. We're going to do the same thing. We are going to draw out meiosis, okay? Except there's two rounds of meiosis. There's meiosis 1 and meiosis 2. So I'm going to draw these using crayons. If you don't have crayons, which is totally cool, then I would use pen and pencil, or maybe 2 different colors of pens. Use whatever you want, but let's get some color. So let's have 2 chromosomes, like such, okay? And I'm going to pick, I don't know, let's pick number 12, okay? If you don't like chromosome number 12, then make it number 1, make it number 5, make it number 16. I have no preference, but pick a number. So here's my homologous pair, number 12. And let's continue with the R's. So here's dominant R and recessive R, okay? So here is your body cell. This is a diploid cell, and it carries 2 copies of each chromosome. Now, each of us carries 2 copies of each chromosome, but where did they come from? Well, we all got our chromosomes from our biological parents. So I'm going to define purple as my biological mother. This is the chromosome that I received from my biological chromosome. Excuse me, from my biological mother. Okay? And then the other chromosome that I carry, this I received from my, if I can spell, there we go, biological father, which I'm going to color as green, like such, okay? So there's my cell. This is going to be a cell that sits inside of my gonad. So this is a cell that sits inside of my ovary if I'm a female, or my testicle if I'm a male, and it's a diploid cell. Now, before this cell can go through division, the cell has to go through a process called DNA replication. Now, to replicate means to copy. So this chromosome is copied. And that's why we see chromosomes in the shape of an X. All this is 2 copies of the same chromosome stuck together. So here's your 2 copies of number 12. This one is big R and big R, 2 copies of the same one. And this is little r, little r. And then I received the big R's, the dominant, from my biological mother, and I received the recessive R's from my biological father. And this is still a diploid cell. This is the cell that enters meiosis. Okay, so we're going to take that cell through meiosis. And you have, of course, the plasma membrane here, and you have the cytoplasm here. And then this in here, this, of course, is the nucleus. All right? So let's go through meiosis. We're going to take that same cell and go through prophase, metaphase, anaphase, telophase. Easy. So the cell itself doesn't look much different, except just like in prophase of mitosis, the nuclear envelope breaks down. So this is going to be what we call prophase 1, implying that this is going to actually happen twice. Okay? So we need our chromosomes. What did we choose? Number 12, okay. Here's my big R's, and here's my lowercase r's. And then we said that the dominant I received from my biological mother, and the recessive I received from my biological father. Boom. Then, just like in mitosis, when we drew this earlier, that's why it's so powerful that we drew mitosis earlier. What do you have here? You have the nuclear envelope, the membrane of the nucleus. It's breaking down because these chromosomes are going to be pulled at 2 opposite sides of the cell. The centrioles separate. Again, this should look a lot like mitosis. The spindle fibers form. And this is still a diploid cell. Looks a lot like mitosis, yes? You bet. So now, what follows prophase is, of course, metaphase. I should actually underline that so it really makes it stand off the page. So here's metaphase, 1. And again, you're dropping this right in your Cornell notes. Now, in mitosis, these chromosomes lined up top to bottom. In metaphase 1, it's different. The chromosomes line up side by side, like such. And so here's my big R, here's my little r, little r. Centrioles are here off to the side. This is number 12. And then now my spindle fibers come in. Watch this. There's only one spindle fiber per chromosome. That's different from mitosis. So the way that students can remember the number of spindle fibers is metaphase 1 only has one spindle fiber per chromosome. Okay, so at this point, here's this term again. Homologous chromosomes, or a same pair, homologous chromosomes line up side by side. Now, that's different. That's different from mitosis. It's a real characteristic that you only find in metaphase 1. So, homologous chromosomes line up side by side, and only one spindle fiber per chromosome. All right, so if that's metaphase, don't forget this is PMAT. Prophase, metaphase, what comes next? Anaphase. What is the classic characteristic of anaphase? That these spindle fibers pull. So now this spindle fiber is going to pull this chromosome this way, and this spindle fiber is going to pull the other chromosome, or the homologous chromosome, the opposite way. This is what allows meiosis to separate or segregate chromosomes. So this is going to be anaphase 1. That's a pretty good circle. And then I always try to angle the chromatids like such to help the students understand that there's pulling here, there's motion here. So these spindle fibers are pulling. It's like grabbing someone's belt and pulling on it, okay? So let's get that centromere there. There's number 12. Here's big R, here's little r, little r. Oh, do you know what I did not do on the previous question? Or on the previous diagram? I did not color coordinate. Let's do that, shall we? There we go. So these are the chromosomes I carry. These are from my biological parents. And I am separating these chromosomes that I received from my biological parents. And I am eventually going to give these to Em. These are the chromosomes that Emily received. Like such. So here in anaphase 1, homologous chromosomes, I'm just, for shorthand, I'm just going to write homochromos. Homologous chromosomes separate. This is the heart and soul of meiosis right there. This is what allows us to separate those alleles. Recall earlier, right? We took the diploid chromosomes and we separated them. During meiosis, what allows the separation? It's all about anaphase 1. Those chromosomes are pulled to opposite sides of the cell. And this is still a diploid cell. Well, then after anaphase is, of course, telophase. Remember that double-sided cell? Telophase 1. Right? Chromosome here. This is number 12. My recessive R's. My dominant R's. My biological mother's chromosomes. These are the chromosomes I received from my biological mother. These are the chromosomes the chromosome I received from my biological father, like this. So what's happening here? Well, there's a lot going on. The plasma membrane pinches in. That's what's happening here. It's called a cleavage furrow as the plasma membrane starts to pull in. All right, the plasma membrane pinches in. The nuclear envelope reforms. The spindle fibers, which I didn't draw, so let's draw those now. There we go. The spindle fibers break down. And this is still a diploid cell because you still have one membrane surrounding the whole cell. Then you get cytokinesis. Cyto, split, process. This is the process that splits the cell. Cytokinesis occurs, and what you end up with now are two separate cells. I'll draw the nucleus nice and big so that you can see chromosome on the inside. So this is referred to as meiosis 1. And as you can see, the whole point of meiosis 1, what's the whole goal here? The whole point of meiosis 1 is to separate homologous chromosomes into two separate cells. Okay? Well, that's meiosis 1. So now what we need to do is go through meiosis 2. And this is even easier. Meiosis 2 is prophase, metaphase, anaphase, telophase all over again. Okay? Except it's only with one chromosome. So what we're addressing here now, this is meiosis 2. See, there's two cell divisions in meiosis. So we call the first one meiosis 1, and we call the second one, ready? Meiosis 2. So, here's your two cells. And of course, this will be prophase 2. And it's going to look very familiar because, see, the characteristics of prophase are always the same. The same thing happens in prophase, whether it's in mitosis, meiosis 1, or meiosis 2. Prophase is always the same characteristics. Da-da-da-da. Draw a little centriole, some spindle fibers in between. Okay, so what happens here? Nuclear envelope breaks down. Centrioles separate. Spindle fibers form. Same characteristics. Why? It's prophase. Okay? Except now, in two separate cells, I have the chromosomes that I receive from my biological mother in one cell and from my biological father in the other cell. And this cell is now haploid. Why? Because it's only half of the chromosome. It's only one copy. Well, prophase is followed by metaphase. Metaphase 2. Well, if you'll recall, in metaphase 1, there was only one spindle fiber per chromosome, whereas in metaphase 2, there's going to be two spindle fibers per chromosome. Okay? So go like that, and then go like that. Need a centriole. Again, this isn't difficult. It takes a little time. This might take us 20 minutes. Okay, no big deal. Watch, two spindle fibers per chromosome. Okay, that's a really big deal. Two spindle fibers per chromosome. Okay, well, if that's metaphase 2, you know what's coming next. What always follows metaphase? Always. Anaphase. So this is anaphase. Anaphase 2, right? And what always happens during anaphase? Always. What happens during anaphase is that spindle fibers pull. Well, if you have two spindle fibers, each pulling on the same chromosome, that X-shaped chromosome, it's two copies of the same chromosome. If one spindle fiber is pulling this way, one chromosome goes to the left and one chromosome goes to the right. All that is two copies of the same chromosome stuck together. So one gets pulled over here and one gets pulled over here. Here's our big R, number 12, number 12. Centriole, centriole. And again, bend them just a little bit to show motion, to imply motion. This is getting pulled this way, and this is getting pulled this way. Like that. It starts like this, and then the X-shaped chromosome gets pulled like this. And then the same thing happens over here. Except with the recessive. Pull and pull. Okay, so here copies of the same chromosome are separated. And of course, haploid cell. Same thing up here. Haploid cell. Okay, so then anaphase pulls them to opposite sides, so that's prophase, metaphase, anaphase. You know what's coming next? Telophase. Same thing, the characteristics of telophase are always the same. So what we're drawing here, this is Meiosis Two. Connect those two. And this is going to be telophase, or telophase, depending on how you want to say it. That's telophase 2. And just like mitosis, and just like meiosis 1, the characteristics of telophase are always the same. That's what makes it telophase. You're not learning anything new here about the phases. You are actually learning what's happening inside the cell with the chromosomes. So 12, 12, 12, put the centriole on the inside, the centromere on the inside. This is dominant R, recessive R, recessive R, the chromosome I received from my biological mother, and the chromosome that I received from my biological father. We are almost there. And of course, what always happens during telophase? Always. Nuclear envelope reforms. Plasma membrane pinches in. Spindle fibers break down. Just classic characteristics of meiosis and mitosis. Classic characteristics of telophase, regardless that it's mitosis or meiosis. And these are, of course, haploid cells. Right? You then get cytokinesis. The process of the cell splitting. This is the process that splits the cell. So this pinches in, and what you're left with are 4 cells. I'll make the nucleus nice and large so that you can see it. Of course, the nucleus really isn't that big. Here's my dominant R, my recessive R. So now, I started with 2 copies of every chromosome. But what am I going to offer Emily? Only 1. I'm only offering Emily one of my chromosomes that I originally received. Okay, so what we have here then, these are gametes. These are germ cells. These are sperm in a male or ova in a female. Okay. These are sex cells. These are the cells that are exchanged during sexual reproduction. Okay, so the whole point of meiosis is to form the cells required for sexual reproduction. So don't forget what we started with. In the very beginning, what we started with was one cell with two chromosomes, right? One for my biological mother, one for my biological father. This is what I carry in all of my cells. This is what you carry in all of your cells. But what do we end up with? We end up with only one chromosome. So meiosis is the separation of homologous chromosomes, and it forms the cells that are used in sexual reproduction. So that took us about 20 minutes, but the drawings themselves are pretty simple, and please understand the major concept, okay? Enjoy.